Bismillahirrahmanirrahim. Assalamu alaikum dear students. Your course is current trends in biochemistry and today's our topic is research report. How to write a research report. You have to give them, you all of them give the report at the end of your semester. So I have to learn you about the research report writing. How you can write a valid research report. First of all, what is a research report? Research report is actually called as a report according to the your research plan which you have made, and how you can present this research in the form of the report or thesis. It is similar as to the thesis of the MPhil or PhD students. In DS, you have to give the report of your research work. You have all done the internship and you have to write up the internship report in the form of your research report. What you are doing in your research, you have a better idea about your research plan. So in this lecture, we will discuss the different components of the important components of the research report, which are very important for writing a valid research report. So you have better idea when you are going through the lecture of the synopsis writing. It is similar to the synopsis writing. Research report is also similar, but we are starting from the components of it: title, introduction, abstract, objectives, methodology, results, discussion, conclusion, references, summary, references. These are the journal components of the research report. In your research report, you have to give a title to your research report. Then you have to give the abstract, which is written at the end of the complete research report have been written. Then the introduction, then the objective parts, and then the methodology. All these components are similar to the synopsis, which you have been learned earlier. These all our components are similar with the synopsis. The new things in your research report is results, discussion, conclusion, summary, and references. References are also present in the synopsis. So you have to go through, or you have a better idea about the results and discussions and the conclusion and summary section. But I have to give you the idea about all these components one by one for writing your research report. Other than these components, some journal components of the research report is also present, which are the acknowledgement, section, declaration section, list of figures, of figures, list of abbreviations, list of tables, list of different tables, acknowledgements, declaration, list of figures, list of abbreviations, list of tables, table of content, in which you give all the sections present in your research report. Funding agency, which funding agency or which research center which give help for your study, your research report to be write up. You have to give the name and the contribution of that agency in your funding agency section. In declaration section, you have to give the declaration of that this is the whole study of your own and you have the copyright of this study. In list of figures, you have to present all the figures list at which page your figures are present. List of abbreviations, you have to give the abbreviations. If you have give any type of abbreviations in your research report, you have to make a list of the abbreviations in your research report. List of tables, how much tables you have presented in your research report, you have to make a list of that tables in your list of tables. In case of acknowledgement, you have to acknowledge all the institutes, all the supervisors, all the students which are involved, which are helpful for your research report writing, for your research. You have to make the acknowledgement in the acknowledgement section for that persons. Now let's start with the different major components of your research report. As we have studied earlier, the title must be very comprehensive, very brief, and it reflects the importance and objectives of your research. So you have to make it at the end of your complete research report writing. After that, you have to give the abstract. In abstract, you summarize the whole your research plan, your research report in a passage which is called as the abstract, in which you have given all the values, all the results. Abstract section also contains some subsections. In this, you have give some lines about intro, 1 to 2 lines about introduction. Then you have to write your objectives in 1 to 2 lines, plan of your research. Then you have to present the methodology which you have adopted for your research plan, for the write-up of your research report. Then after that, 2 to 3 lines. Then you have to write up about the results. Results are actually the findings of your research which you have planned. Your research findings. In 2 to 3 lines. Then you have to give the conclusion of your research. What you find in your research, you have to give the idea about the research planning of your research in the conclusion. In this way, you can write a valid and a very good learner's very informative abstract. After the writing of abstract, the next section is about the introduction section. In this introduction section, as we studied earlier in the synopsis lecture, we must give the important feature of our research plan. We give the idea about the importance of our research and give the background of our research. That major points of research which give you the idea to plan your research report according to that criteria, according to that said part of your research. If you have planned your research about the antioxidants or for the enzymes, you have to give the idea in your introduction about how much this antioxidant or enzyme is important, how this enzyme is present in by which concentration is present in that, and how can you, the others one, reported this in their research reports. In the next section, which is the review of literature. As we studied earlier, review of literature is actually review of all the literature which we have studied for the plan of our research report. If you have 20, 30 literature articles, you have to write the review of that articles about your topic, your specific topic on which you write your report. You must have to explain all your literature survey in your review of literature in comprehensive way. It is similar to the introductory part, but it is more elaborated, it is more comprehensive as compared to introduction. In this review of literature, you have to write about all the importance of your research report. After review of literature, you have to write the methodology. In this section, you have to give the methods and materials which are required, which you have used, and the methods which you have used for your research to be going on. For example, if you have studied the different antioxidants in your sample, you have to give the methods of each antioxidant separately. If you have studied the catalase enzyme, TOD enzyme, SOD enzyme, so on, you have to give the method of each one in detailed form and give the citation of which method you have been used for the measurement of the catalase amount in your sample, peroxidase amount in your sample, and superoxide dismutase amount in your sample, or so on. If you study the different medical enzymes, like you have studied the amount of alkaline phosphatase, or if you have studied the amount of glucose present in the samples of your patients, you have to give the method by which method you have calculated the amount of glucose present in the sample. So you have give the complete methodology of your research, what you have done in this section. Also you have give the idea about the instruments which you have choose for doing all these type of methods. You have get the better idea when you have gone through the synopsis section, synopsis lecture, in which I have give you the idea about the material and method section more briefly. Okay, next we have shifted up to the section of results. Different types of results we have presented. This is actually the interpretation of your data. Whatever your data is interpreted, if you have interpreted your data in a very well manner, your research report would be a miracle. Your results show the novelty of your work. If you have present your results in a very effective way, or it is effective on the readers, on the other persons, they would be the success of your research report. You have get the calculated amounts of different biomolecules. For example, glucose. You have measured glucose, you have measured protein in your sample, you have measured different enzymes in your samples. You must have the standard curves of all these things. What are the standard curves? Standard curves are actually you must learn about the standard curves. When you calculated the amount of the unknown sample, you have to run the standards with this unknown sample. If you have to calculate the amount of glucose in your samples, you must have different standards of the glucose concentration, and by putting the values of all these concentration and add this the absorbance. You have taken the absorbance by using the spectrophotometer for the calculation of your in your sample the amount of the glucose, for example. So you have to give the concentration standards which have the given concentration 0.5 grams, for example 10, 1 gram, 1 gram, 1.5 gram, 2 gram, 2.5 and 3 grams per your calculated value. Then you have some absorbance of these standards. 0.5 gives the 0.2, 0.4, 0.6. I will give you the idea about the standard curve. On the x-axis, you have put on the values of your concentration 0.5, 1, 1.5, 2, 2.5, 3, 3.5, for example. These are different standards which you have chosen. And here, you at the y-axis, you give the absorbance of your sample. 0.2, 0.4, 0.6, 0.8, 1, 1.2, 1.4, so on. Here this sample have absorbance at 0.37. This sample has 0.5. This has 0.7. This has 0.85. This has the value at 1.1. So these are your standard. This is your standard curve. Now you have the unknown sample of your sample. If it has the absorbance, it shows the absorbance at 0.9. You draw a line on this. From this on the standard curve and here. Now you can get the concentration by using this standard curve, the unknown samples concentration by using this standard curve. So always when you presented your results, you must have the idea about the standard curves. You must have run the standards or blanks with your sample for calculating the exact amount of in your samples. These are the different types of results. Which results you get, you have to elaborate them in your result section. In result section, you give the different graphs of your results, figures of your results. If it is a gel, you have gone through the polyacrylamide gel electrophoresis and you have get the picture of your gel. You can present in the result section. After this, the next section is the discussion section. Discussion section is very important because you have give all the results, but if you have not discussed them with the previous ones, in the discussion section, you have to compare your results with the results of the other authors, with the results of the other scientists. You have in the discussion section, you compare your results with the other scientists' results. These are found out in your literature, which you have already done. Compare your results with the results of the other scientists. By comparing them and putting if you have got that the glucose level is high in your sample and the other one also reported that the glucose level is high in their sample, so you can report them in the discussion section by citing the article of that person. Similarly, if the antioxidants level is high in your sample and other scientists, other authors also reported that the level of antioxidants are high in their sample, so you can report the same results are compared with that. But this is not that if your concentration you don't think of that, always the positive results are the results, the negative results are also the results, and you can represent them in your report. For example, if the level of your antioxidant is lower down, you can compare it with the others. Maybe other ones are also reported the low level of antioxidants in their samples, so you can report them also. Then after discussion, when you complete the whole discussion of all the results which you have presented in your report, then you have give the final conclusion of your research. You have hypothesized something in your research plan, and you have now have the findings about that hypothesis. So you have give the idea about the conclusion. In conclusion, you give the idea about what your results, your findings are important, and why the other ones can choose this type of research for future research. How can they plan their future research? Also in the conclusion section, you have give the future prospects. In future, which type of research can be planned according to your research report? So you have to give in your conclusion section what you have concluded by the results which you have found. After the conclusion section, you have give the summary. This is of only 5 to 6 lines maximum. In summary, which is of 1 page, you shortly report all your plan and your findings with concluding remarks. In the summary, which is of 1 page, shortly report all your plan which you have planned for your research report and your findings which you have get in the form of results and conclusion with concluding remarks. What you have concluded, what you have conclude from these results or these plan, you have to be summarized in the summary section. After summary section, you have give the last part of your research report is the reference list, in which you list all the references either you used in introduction section, either you use in your methodology section, or either you use in your review of literature section, either you use in your discussion section. All the references, all the citations which you have given in your research report, you have to be made a complete list of that references in the your reference list in the your references section. These all are the parts of the research report. When you write a complete research report, you have to check the level of plagiarism in your research report, which must be less than 20%. It must be less than 20%. So, if you have write by your own and in your own words, you have get a very better report first time, that is less than 20%. But if you have only copy paste, then it has the plagiarism, so you have to remove it. This is all about the research report writing. If you have any questions regarding this research report lecture, you can approach me on my WhatsApp group, either on my email address or by putting a comment in the comment section. I hope you have a better idea now about the writing of a research report. Thank you so very much, students. Allah Hafiz.